Good morning, everybody. Thank you for joining us today. My name is Rhonda Henshaw-Powell. I'll be your moderator for the day. Please make sure that you remain on mute. Check your audio right now. We have a very exciting presentation for you in this next in our Knowledge Resource Series sessions for you. I would like to hand over to Dr. Pupak Farahani. She is our host for today. Thank you very much, Pupak.
Thank you very much. Hello, everyone. My name is Pupak Farahani, Marketing Manager at Advanced Cell Diagnostics, and your host for today. Welcome to ACD's Knowledge Resources webinar series, where we'll give you a closer understanding of how our RNASCOPE technology is contributing to specific application areas. We begin today's knowledge series on the topic of spatial genomics, presented by Dr. Anushka Dikshit. Dr. Anushka Dikshit is an application scientist with Advanced Cell Diagnostics with background in cancer research and immuno-oncology. She's focused on expanding the application of RNASCOPE technology for oncology research. Prior to working for ACD, Anushka was involved in identifying novel therapeutic targets for melanoma as a postdoctoral fellow at Duke University. She obtained her doctoral degree from Southern Illinois University School of Medicine in molecular physiology. With that, we will get started.
Thank you for that introduction, Pupak. As Pupak mentioned, today I'll give examples to detail the use of RNASCOPE in situ hybridization technology in spatial genomics and help you better understand how you can apply this technology for your research. Let's start by understanding what encompasses the RNASCOPE in situ hybridization technology. For those of you who are not familiar with this assay, it's an in situ hybridization platform that was designed to overcome some of the traditional limitations of an ISH assay while still providing really high sensitivity and specificity of detection, while overcoming some of the traditional limitations like background and signal amplification. The RNASCOPE technology is an ideal spatial analysis solution to interrogate really complex tissues. It is extremely specific and sensitive method to detect RNA targets, including RNA biomarkers in cells and tissues, while maintaining the morphological context and providing detection at single cell resolution. It mainly consists of three parts: the unique probe design strategy, where an ACD will design a target probe against the sequence of your interest, a signal amplification system that generates high signal to noise ratio, and finally, a visualization system that allows you to see a single RNA molecule as a punctate dot, either in a chromogenic or fluorescent platform. The assay allows for spatial mapping of your mRNA targets, and these include not just your mRNA, but long non-coding RNAs, splice variants, homologous sequences, point mutations, and so forth. And we allow detection of your targets of interest in a wide variety of sample types as well, including FFPE tissues, fixed and fresh frozen tissues, cultured cells, and PBMCs. So as you can see, our assay allows for really wide range of flexibility in which you can apply this to your research. In the next couple of slides, I want to go in a little bit more detail to help you understand what makes our assay so sensitive and specific and lets it stand apart from some of the other tools that are out there. As I mentioned, the two key features of our technology are the probe design strategy and signal amplification system. In this particular screen, we have depicted the RNASCOPE ISH probe as a Z molecule to highlight the different parts of the probe, which include the target binding site, which, as the name suggests, is a oligo sequence designed to be complementary to your target sequence of interest. The preamplifier binding site forms the foundation of amplification tree development. When we design our probes, we design them as pooled probes such that the two Z probes have to bind in tandem next to each other on the target sequence. This allows for binding of the preamplifier on the Z pair After which you have the amplifiers that bind on to the preamplifier, followed by which either the chromogenically or fluorescently labeled probes bind on to the tree. This amplification system ensures high sensitivity of detection, allowing visualization of even the low expressing targets. Finally, as I mentioned, the preamplifier will not bind on to the Z probes unless they are bound in tandem. So if a Z probe non-specifically binds to a sequence, then the preamplifier is not able to bind to the probes and no amplification tree is formed. As a result, no signal is detected. This two strategies ensures very high sensitivity and specificity of detection. Whenever we have new customers running the RNAscope assay, we always encourage them to run controls, such as negative and positive controls along with their assay. And this actually also extends to established customers because it is important to understand the signal you're visualizing is real and is not background. So we always encourage running both positive and negative controls. Here is an example of our negative control probe DAG-P, which is a target in which is a bacterial gene and shows no signal in a mammalian tissue like the human lung. On the other hand, our positive control probe is PPIB, which is ubiquitously expressed in the mammalian tissue. So this gives you an idea of the difference in the signal of when you see a positive RNAscope signal versus a negative signal. A positive signal is a clear punctate dot as indicated here, whereas no negative control should not show any distinct signal. On the other hand, you would usually expect your target probe to show a signal somewhere in between your positive and negative control, wherein you will be able to assess specifically where your target is expressing. We also allow for quantification of the RNAscope signal, and we allow for two different methods, the semi-quantitative method and the quantitative method. In the semi-quantitative analysis, we use a scoring system where based on the degree of expression of your target, you can score your staining. For example, if you have 1 to 3 dots per cell, then you will give a score of 1. If you have around 4 to 9 dots per cell, then you'll give a score of 2, and so forth. This allows for really nice relative comparison of your signal expression between different samples. On the other hand, if you do want to perform extensive quantitative analysis, we allow for that as well, wherein we have partnered with Indica Labs, who have the Halo software that can be used efficiently to segment your tissue and analyze staining patterns to provide an actual quantification of your RNAscope staining on the tissue of your interest. So based on your research requirements, you can go either the semi-quantitative or quantitative way of RNAscope signal analysis. Broadly, the RNAscope spatial mapping technology encompasses 3 different assays. The RNAscope assay, which was designed for targets that are over 300 nucleotides long. The BASESCOPE assay that was designed for targets that are between 50 to 300 nucleotides long, and these include your splice variants, homologous sequences, and point mutations. And finally, our newest assay that was launched this summer, the microRNAscope assay, is designed for really short RNA targets that are between 17 to 15 nucleotides long. And these include your microRNAs, siRNAs, and antisense oligos. So based on the target of interest and your research requirements, you can select either the RNAscope, BASESCOPE, or microRNAscope assay for your research. In this slide, I just wanted to describe a broad overview, give you a broad overview of our product portfolio. Here we have described all the different assays in all the different configurations that we make available. As you can see, we are very flexible with respect to the plexing capabilities that we offer. For RNAscope, we offer single plex, 2-plex, and up to 12-plex, wherein you can visualize 12 targets on the same section of the tissue. For BASESCOPE, we have up to duplex, and for microRNAscope, currently we offer singleplex detection of RNA targets. A lot of these assays are available both in chromogenic and fluorescent platforms, and we also make these assays available in manual and automated platforms. So based on your capabilities and infrastructure, you can select the assay that suits best for your research. Currently, we have over 26,000 probes in our catalog, where we already have these pre-designed probes ready for you. But if you want to design a probe against a target that's not in our catalog, we can very easily design any probe against any gene in any species in as less than as 2 weeks. So do refer to our catalog to see what genes we already have probes for. Finally, the focus of today's presentation is to give everyone an overview of how well adopted this technology is and the broad range of applications that it offers. So I'm going to talk to you about the applications of the RNAscope technology in basic science research, in infectious disease research, in biopharma discovery and development, and cancer research. Because our assay allows for flexibility in sample type and in assay type, we are well positioned in identifying or detecting targets such as splice variants, non-coding RNAs, for targets such as GPCRs, which are known to have really poor antibodies, or for multiplexing for validating single-cell RNA sequencing, and so forth. So by giving you examples for each of these targets in these different applications areas, I hope to expand your understanding of how this spatial mapping technology can be applied for your research. To begin with, let's start with our first application, that is to understand how RNAscope has been used in neuroscience research. In the field of neuroscience, it becomes extremely important to understand that as you look at different sections of the brain, the gene expression can vary dramatically. And so it's important to use a technology that is extremely precise in mapping gene expression. RNAscope has been widely used for detecting ion channels, GPCRs in the brain, for visualizing cellular activity in the neurons, for visualizing different neuronal subpopulations in the brain, and for studying the expression of splice variants like circular RNAs, which play an important role in developing mouse and human brains. So to start with, I want to give you an example of how RNAscope has been used to study the different subpopulations of neurons in this particular example. A paper published by Koke et al. Described the different subpopulations of median spiny neurons and identified gene signatures for each of these subtypes. So we based this project on that paper, and we selected genes specific for D1 and a set of genes specific for D2 medium spiny neurons, and wanted to visualize both these D1 and D2 populations simultaneously on the same section of the brain. So we designed RNAscope probes specific for these targets and performed the HYPLEX assay and visualized all the 12 targets on the same section. The idea was to spatially resolve the expression of these targets and visualize these different subpopulations of neurons simultaneously. So this is an image or a GIF that we created from the HYPLEX image registration software, which we provide along with our HYPLEX assay, wherein you can visualize all the 12 targets simultaneously and understand how these subpopulations of neurons are spatially oriented with respect to each other in these specific regions of the brain. In this particular example, we are visualizing the mouse brain striatum and resolving the D1 and D2 populations of MSN neurons. Ultimately, when you use our image registration software, this is how your image will appear if you decide to overlay all 12 targets simultaneously. But we have the capability of toggling genes on and off based on your target of interest and studying expression of individual genes based on which genes you want to focus on. So this really allows you to study gene expression for individual subpopulation of neurons and better understand their function. So with that, I want to move on to an example which shows you the importance of multiplexing for spatial mapping. Here is an example wherein these researchers published this paper in Cell Reports in 2019, and they were trying to establish a cell atlas for vagal sensory neurons. They utilized single-cell RNA sequencing analysis to identify different subpopulations of vagal sensory neurons, and then designed probes against markers specific for these subpopulations because they wanted to validate and visualize these subpopulations using RNAscope. So they ended up using 25 different RNAscope probes to visualize these subpopulations in situ. Now, by using a different combination of probes, they were able to co-localize these markers and identify these subpopulations, but they ended up utilizing a lot of mouse brain sections to perform this particular RNAscope analysis. If they had the HYPLEX assay, they would have been able to utilize significantly less amount of sample and get the same data by conserving a significant amount of their sample. So this is a great example of how multiplexing can be an extremely important tool, especially in neuroscience, where samples are normally scarce and expression patterns vary dramatically as you cut through sections of the brain. In another example, this particular paper published again in Cell Reports was studying expression of PRDM12, which is an important target that regulates the development of nociceptive neurons. So again, an application of RNAscope in neurogenesis using RNAscope. In this particular example, they have utilized the dual RNAscope-IF technology, wherein they used a workflow where they combined RNAscope with immunofluorescence. This allowed them to visualize RNA target along with a protein target on the same section of the tissue. So they wanted to exactly study which cells were expressing this PRDM12 that was important in development and maintenance of the nociceptive neurons. As you can see here, they co-localized the PRDM12 with TRK-A, C, and B antibodies and analyzed that PRDM12 co-localized nicely with the TRK-A positive neurons, but not so much with TRK-C or TRK-B positive neurons. Finally, they wanted to understand the role of this target in homeostasis of nociceptive neurons, and they utilized the RNAscope multiplex assay to demonstrate that targets such as neurogenin 1 and 2, which are important in maintenance and neurogenesis of nociceptive neurons, are regulated by PRDM12. As you can see here, all three targets are co-expressed in this particular embryonic brain section, demonstrating the nociceptive neurons co-express all these targets. And upon knockdown of PRDM12, what they were able to show is that the expression of neurogenin 1 and 2 is lost in the absence of PRDM12, highlighting the role of this target in neurogenesis and homeostasis of nociceptive neurons. Next, I want to bring your attention to a very seminal paper that was published in Nature last year, where they utilized single nuclear RNA sequencing to study the different subpopulations of oligodendrocytes in MS brain. MS, as you know, is a disorder caused by demyelination of neurons and which leads to neurodegenerative disorder. In this particular example, these researchers have compared a normal brain to a MS patient brain from postmortem samples, and what they were able to show using the BASESCOPE assay was that there was differential expression in the oligodendrocyte population between these two neurons. Using probes against specific markers, they were able to show that oligo 5 specific marker KLK6 was independently expressed compared to oligo 1 specific marker CDH20, showing that these are independent markers that are exclusively expressed in different subpopulation of oligodendrocytes. They went a step further in showing that the oligodendrocyte population is distinct between a normal brain and an MS brain. And in the MS brain, they were able to identify a hybrid cell called oligodendroclea, which showed characteristics of oligodendrocytes and microglia. And here, they're using the BASESCOPE assay, they have been able to co-localize oligodendrocyte marker OLIG1-2 with CD74. And this indicates that this particular cell showing co-localization is an oligodendroclea, and expression of this particular cell type is higher in MS lesions compared to normal brain. So this particular hybrid cell might be potentiating the MS pathology in this particular disease. Finally, microRNAs are known to be important regulators of gene expression in the developing and in adult brain. And using our new microRNAscope assay, we are now able to visualize these important targets in the brain. And here's an example of how we have used our microRNAscope assay to visualize all these different targets in the mouse brain. Wherein, for example, you can see MIR138 specifically expressed here in the Purkinje cells of the mouse brain. So with that, I want to move on to our second application for today, which is the application of RNAscope in cancer research. Now, it is important to understand that tumors are extremely heterogeneous tissues, which comprise of different cell types, including your cancer cells, endothelial cells, fibroblasts, and so on, all engulfed in the extracellular matrix. So to really understand how these different cell types are interacting with each other, it is important to use a precise transcriptomic tool that will help us identify targets and predictive biomarkers to further our therapeutic strategies. So we believe that RNAscope is an ideal tool since it allows for visualizing tumor heterogeneity, for detecting immune subtypes, for visualizing TCRs on T cells, identifying splice variants, and detecting engineered immune cells. So with this broad application in cancer research, I want to start with helping you understand how this really sensitive technology can help identify and characterize specialized immune cells. So in this project, we wanted to visualize specialized immune cells like NK and DC cells, mainly to show the correlation of NK cells with conventional DC cells 1. So from this paper published from Bocher in Cell, they were able to show that in a tumor microenvironment, NK cells can release soluble factors like CCL5 and XCL1, which recruit DC cells like conventional DC1 type cells, which possess the markers for these soluble factors. And these DC cells ultimately recruit cytotoxic T lymphocytes from the draining lymph nodes. And these cytotoxic T lymphocytes can ultimately mount an anti-tumor immune response in the tumor microenvironment. So we wanted to show that using RNAscope technology, you can actually identify and visualize these specialized immune cell populations. So we designed probes against specific markers belonging to NK cells and against markers belonging to the soluble factors secreted by NK cells like XCL1 and CCL5. So here we show that we were able to co-localize all these 4 markers to identify NK cells in this cervical cancer tumor. And compared to sample 1, sample 3 showed low to no presence of these NK cells. So we were able to co-localize all 4 markers to identify these cells that are not very abundantly present in the tumor microenvironment. And this is the strength of our technology because it allows you to go down to that single cell resolution and identify these specialized immune cells that are not abundantly present in tumors, but might hold the key to understanding how anti-tumor immune response can be enhanced. Next, we wanted to see if these NK cells are recruiting any DC cells to the tumor sites. So again, we use specific probes against specific markers for conventional dendritic cell type 1. These include THPD, CLEC9A, XCR1, and CCR5. Again, to highlight, XCR1 and CCR5 are receptors to the soluble factors secreted by NK cells, and we were able to co-localize all 4 markers on specific cells, indicating that these are subsets of the DC cells that we're interested in and show correlation with the presence of NK cells. Again, in sample 3, we were not able to see as many DC cells compared to sample 1, showing correlation between NK and DC cells. And finally, we compared T cell infiltration to the presence of NK and DC cells, and we were able to show that indeed sample 1 that showed a presence of these specialized NK and DC cells showed higher degree of CD8 positive T cell infiltration compared to sample 3. So this allowed us to very specifically interrogate a niche in this cervical cancer tumor to show that the NK and DC cells were getting recruited to the same part of the tumor, and this also showed increased infiltration of CD8 positive T cells. So this type of specific analysis can be very easily done using the RNASCOPE technology, since it allows you to specifically co-localize multiple markers to identify specialized immune cells. Next, I want to bring your attention to how important it is to characterize tumor heterogeneity and how RNASCOPE can help with intracellular and intercellular heterogeneity analysis. In this example, we are visualizing PD-L1 expression in different lung cancer tumors. And as you can see, different lung cancer tumors express different degree of PD-L1, showing intertumor heterogeneity. But interestingly, when we visualize PD-L1 in the same lung cancer tumor, we saw that based on the region of the tumor, the expression of this marker can vary dramatically as well. And this type of sensitive detection can play an important role in determining therapeutic resistance for certain malignancies. Here's an example where we visualized multiple checkpoint markers using RNASCOPE in different lung cancer tumors. And based on the expression of these checkpoint markers, we labeled the top row tumor as a hot core and the bottom row tumor as cold core, since it showed very low expression of these checkpoint markers. When we decided to compare immune cell infiltration between these tumors, it was no surprise that we found high degree of immune cell infiltration in the hot core tumor compared to the cold core tumor. And these included markers like interferon gamma, CD8, FOXP3, CD4, which are markers of T cells, and other markers like CXCL10, CCL22, CD68, and CD163, which are markers of M1 and M2 macrophages. So what we were able to show was tumors that had high expression of checkpoint markers also showed high expression of these immune cell markers, indicating increased immune cell infiltration. And what this shows is that this particular tumor potentially has a better rate of response to immune checkpoint inhibitors compared to the cold tumor. An important thing again to highlight here is that RNASCOPE allows you to identify the cellular source of secreted factors like chemokines and cytokines that is not possible by protein-based methods like IHC and IF. So ultimately, this helped us understand immune cell infiltration was significantly higher in the hot core tumor compared to the cold core tumor. Next, I want to briefly touch upon a very interesting collaboration between ACD, Bio-Techne, and NanoString Technologies, where RNASCOPE is now a part of the GeoMX DSP workflow, where RNASCOPE is used as a molecular guide to spatially map GeoMX data. So essentially, you would use RNASCOPE probes to identify regions of interest in a complex tissue sample. Then, based on the RNAscope expression, you will select specific regions within the tumor to profile using GEOMX DSP. After you've profiled those specific regions, you can validate those data again by using RNAscope. So here's how the workflow looks. You identify targets of interest and probe them on your tumor. Based on the expression pattern, you will illuminate specific regions within the tumor using the DSP, and only these regions will be profiled for gene expression analysis, and based on the expression pattern, they can be further validated using RNAscope. So in this particular example, we used PD1, PD-L1, and CD3 RNAscope probes to interrogate this lung cancer tissue. And we saw that certain regions showed high expression of PD1 and CD3 that were high stromal regions, while certain regions showed low expression of markers like CD3 and PD1. And there were certain regions that were high in tumor cells, showed high expression of PD-L1. So we selected a good representation of all these different ROIs and profiled using the DSP, which allowed us to get gene expression information for over 1,800 targets. After which we were able to do analysis wherein we were able to show differential expression of all these targets based on our expression of PD-L1 or PD-1. For example, targets that showed upregulation in regions enriched in PD-L1 were drastically different than targets that showed enrichment where PD-L1 was low. So these type of analysis really help you interrogate specifically which regions of the tissue you want to study and understand gene expression profiles in those particular areas. So to make things easier, we have validated a panel of probe combinations wherein we have tested these probes for you, and these include a wide range of targets that will profile immune checkpoint markers, T cells, macrophages, and so forth. Having said that, these are just a few targets that we've validated in-house, but you could use any marker for your molecular, for RNAscope to molecularly guide GEOMX DSP to perform this joint workflow. And if you want more information about how you can combine these technologies and use the joint workflow, please visit our website or reach out to us and we'll be happy to provide you more information. Next, I want to bring your attention to the BASESCOPE assay, which is a critical tool to analyze splice variants and point mutations. In this particular example, these researchers utilized the BASESCOPE assay to identify EGFR point mutations in this lung cancer tumor. They wanted to use the BASESCOPE technology to stratify patients for TKI treatment. Now, EGFR mutations such as EGFR L858R are known to respond well to TKI therapy. On the other hand, patients with T790M mutations do not respond well to TKI treatments. So using the BASESCOPE assay, they were able to identify patients with this particular mutation and stratify them for TKI treatment. They also went a step ahead and used PCR to show that the BASESCOPE data was comparable to what they would obtain by PCR. And this helped them understand that BASESCOPE is not only sensitive and specific, but it provided the spatial and morphological context that was not able, that they were not able to obtain by traditional techniques like PCR. So the specificity of BASESCOPE assay was validated by PCR, and this allowed us to study the morphology of the tumors and stratify patients for the necessary treatment. So with that, I want to change tracks a little bit and talk to you about a very exciting application of RNAscope, that is use of RNAscope in cell and gene therapies. To start off, I want to introduce a joint study that ACD did with Bluebird Bio, where Bluebird Bio was developing CAR T therapies for solid tumors. And specifically, they were developing therapies for these anti-BCMA and anti-ROR1 treatments. Using RNASCOPE technology, we were able to design specific probes against the CAR vectors that they were using in their CAR T cell therapy. In this particular example, the mice were injected with anti-BCMA CAR T cells, and we are now visualizing the expression of the CAR T cells and the immune response that was mounted by these CAR T cells using RNASCOPE. As you can see, this xenograft is expressing the BCMA antigen, and in the presence of the BCMA antigen, the anti-BCMA CAR T cells engage, which is indicated by positive granzyme B staining. And the CAR T cells are indicated by in the red staining because we've designed a specific probe against the CAR 3' UTR of the CAR vector. Here we have used the same tissue but performed multiplexing to show not only the granzyme B and the CAR T cells, but also to combine them with markers like interferon gamma and CD3, which help us to interrogate and differentiate between endogenous T cells, CAR T cells, and show engagement of these CAR T cells in presence of the BCMA antigen. On the other hand, their second therapy, which was the anti-ROR1 therapy, was not as successful. They were starting to see some toxicity in the lung and liver, and when they analyzed for expression of ROR1 using IFC, they were not able to see any antigen, but when they used RNAscope to interrogate expression of ROR1, they saw that there was some basal expression of ROR1 in the lungs, which was causing the pulmonary toxicity due to the ROR1 CAR T therapy. So this helped them understand that the BCMA therapy worked really well and the ROR1 therapy was not specific, or rather showed on target but off tumor effects and was therefore toxic. So here's an indication of how well these therapies work. When these were injected in the xenograft mouse model, the anti-ROR1 CAR T therapy did not work as well in reducing the tumor burden, but the anti-BCMA CAR T therapy worked really well and showed tumor regression. Another example of how the BASESCOPE assay was used to study AAV therapy is where these particular researchers utilize BASESCOPE to identify their transgene and their AAV vector. So in gene therapy studies, it is important to understand how well the gene therapy has been delivered to the target tissue and the target cell of interest, and to study the expression of the transgene to understand if the vector is expressing the transgene at the intended degree. So in this particular example, we designed probes, BASESCOPE probes, against the AAV vector and the transgene, and utilized our BASESCOPE duplex assay to study the delivery of this particular therapy. So compared to the vehicle, you can see that the transgene and the AAV vector show strong expression in these cells. In red is the AAV transgene mRNA, and in green is the AAV vector genome. What you can appreciate is the subcellular staining or the subcellular resolution that we can observe with this particular analysis, where the AAV vector genome staining can only be observed in the nucleus, as you would expect, but the transgene staining is observed both in the nucleus and the cytoplasm. And here's a quantification of vehicle-treated versus AAV-treated staining, indicating that the delivery was successful, and we were able to see both the transgene and the vector in the intended cells in these liver samples. Next, I want to show you how our newest microRNAscope assay has also been utilized for certain gene therapy strategies, and mainly for interrogating RNAi and antisense oligo therapies. So synthetic oligo-based therapies are showing good promise in preclinical and phase trials, and these include your siRNA and ASO therapies. These are mainly developed for pathologies or targets that are called undruggable or hard to treat. And these include several pathological conditions in oncology research, neuroscience, and autoimmune disorders. For siRNA-based therapies, there are a lot of therapeutic strategies that have been developed to treat cancer, especially target oncogenes that promote malignant transformation or cancer progression. In neuroscience, specifically to treat targets associated with acute brain injuries, and in immunology to mitigate autoimmune disease progression. Similarly, antisense oligo-based therapies have been developed for mitigating expression of cancer-promoting genes, for targeting inaccessible drug targets in neuroscience research, and again for autoimmune therapies in immunology research. So to understand if these therapies are targeting intended genes and to study their delivery in the intended tissue and cell type, it is important that we have a tool to precisely assess the efficacy of these therapies, and microRNAscope assay provides this tool to study these specific ASO and siRNA therapies in tissues. So in this particular example, we had designed, the customers had designed two different siRNA-based therapies, siRNA1 and siRNA2, and we utilized our microRNAscope assay to visualize the delivery of these therapies in the target tissue. So here we have used probe specifically against siRNA1 target, and as you can see that we can specifically detect the siRNA delivered in this tissue, but not in the siRNA2 delivered tissue, indicating the specificity of our assay in identifying siRNA. Similarly, for ASOs in this example, this ASO therapy was delivered in different muscle tissues of this mouse, and with using our microRNAscope assay and designing a specific probe, we were able to identify and visualize these ASO targets specifically in the treated tissue compared to the vehicle tissue. This again highlights that ASO delivery is successful, and we have a tool to precisely analyze this particular delivery in our microRNAscope assay. With that, I want to jump back into one of our last sections for today, which is the application of RNAscope technology in stem cell and developmental biology research. In this particular example, Ayaz et al. Were wanting to analyze expression of certain targets that are important in stem cell renewal, and they were studying the intestinal stem cells and target associated with regeneration of intestinal stem cells. So LGR5 is a very well-known target that is known to be expressed in the intestinal stem cells, and they're responsible for the turnover of the intestinal epithelium. But in this particular study, they were able to identify another population of stem cells that was not LGR5 positive, but positive for a target called clusterin that was also responsible for the turnover or regeneration of intestinal stem cells. And using single cell RNA sequencing, they were able to identify these subpopulation of stem cells, and using RNAscope, they were able to visualize and validate the expression of these stem cells. So here, as you can see, in a normal intestinal epithelium, they were able to identify LGR5 positive and clusterin positive population of stem cells in the crypts of the intestines. As you can see, they are independently expressed, indicating they are independent populations of stem cells. But upon irradiation, that is injury of the intestinal stem cells, what they observed was that the expression of clusterin in this particular tissue dramatically increased upon injury, indicating that expansion or renewal of injury-associated intestinal epithelium might be driven by the clusterin-positive stem cells. As you can see, post-irradiation day 3, the expression of clusterin is significantly higher compared to LGR5. Next, I want to again highlight the utility of our microRNAscope assay in studying the expression of microRNAs important in developmental biology. MicroRNA such as MIR124 is a significant microRNA that regulates gene expression in the developing embryo, especially in the developing brain. As you can see in this beautiful staining, expression of microRNA is predominantly localized in the brain in this particular embryo compared to scramble control. So using our assay, you can interrogate these target microRNAs that are essential and crucial for a developing embryo. Similarly, microRNA such as MIR140 is important in cartilage development of the embryo, and you can see the expression of this microRNA in digits and somites, also in the ear cartilage. And compared to the scramble control, we see a nice signal here in the MIR140 probed sample. So again, our microRNAscope assay now has the capability to interrogate these microRNA targets important for cartilage development as well. Similarly, MIR182, which is an important target in developing embryo, especially in the developing retina. And here's an example of a mouse retina where we see precise expression of this MIR182 interrogated by our microRNAscope. So these are targets that are important regulatory molecules that regulate the expression of other genes, which are important in developmental biology. And with our microRNAscope assay, we can now interrogate the expression of these microRNAs in developing embryos. So with that, I want to come to my final section for today, which is the application of RNAscope in infectious disease research. As I mentioned earlier, RNAscope technology has really helped advance the field of not only cancer research, neuroscience, and developmental biology, but is an important tool, especially in infectious disease research, mainly because with RNAscope technology, probe design strategy is pretty rapid, and we are able to design target probes against any emerging pathogens to help aid and expedite research. A good example of this is the current situation we are in, wherein RNAscope was used extensively in a lot of seminal publications that came out to understand how the SARS-CoV-2 virus infects the host tissue. The first example I want to describe is that of RNAscope, how RNAscope was used to study Zika virus infection. In this particular paper that was published by the CDC, they wanted to study the presence of Zika virus in a host infected, in Zika virus infected patient, and show association of Zika infection with congenital microcephaly and pregnancy loss. So they were studying Zika infected placenta and infant cerebral cortex, and using RNAscope wanted to visualize the presence of the Zika virus in these tissues. So they designed a probe against the Zika virus, and they also designed a probe against the intermediate strand, that is the intermediate replicative strand of the Zika virus, which helped them study the actively replicating virus in this particular infected tissue. So as you can see, both the genomic RNA and intermediate RNA show strong positive signal in these particular tissues, again showing correlation of viral infection with microcephaly and pregnancy loss. So this paper really helped them establish their hypothesis that Zika infection indeed is associated with pregnancy loss and microcephaly. And here's an example from one of the recent papers published in Cell, where they were studying the SARS-CoV-2 virus and showing the infectivity of this virus in host cell tissue. In this particular example, they have used the RNAscope multiplex and singleplex assay to study expression of host cell receptors like ACE2 and proteases like TMPRSS to show a gradient of expression of these markers in our respiratory tract. In this particular example, they have used cytospin samples to show expression of ACE2 receptor and co-localize it with other markers of the bronchi and other lung cells. On the other hand, they used ISH assay to design probe against the ACE2 and TMPRSS. They used the ISH probe against ACE2 and TMPRSS protease to show expression of these markers in different parts of the lung tissue, including nasal epithelium, trachea, and bronchi. And what was interesting was the expression of both these markers was highest in the nasal epithelium, and the expression reduced as they went down in the respiratory tract. What they were also able to show in this particular paper was individuals with cystic fibrosis showed increased risk of SARS-CoV-2 and showed a more serious pathology upon infection with this virus. And what they were able to show was one of the reasons for this could be the expression of ACE2 and TMPRSS, which are important for the virus to infect a host cell, are significantly higher in cystic fibrosis lung compared to normal lung, as you can see here. These nice punctate dots indicate expression of this protease, which is significantly higher in the cystic fibrosis bronchioles compared to the normal lung. So this could be one of the reasons why individuals with existing lung pathologies show a more serious manifestation of this disease. So this is just one of the many papers that have used RNAscope probes to interrogate and study the infectivity of SARS-CoV-2 infection, and we are very proud to contribute to this research. So with that, I want to highlight how well this technology has been adopted over the few years, over the last 10 years, and we have over 2,800 publications as of now, and we have been used extensively, as I mentioned, in different research areas, including basic science research, clinical research, to assess gene and cell therapies, and so forth. So I hope what I've been able to show you today is that the RNAscope technology is a broad umbrella of three major assays: BASESCOPE assay, RNAscope assay, and microRNAscope assay, which will help you interrogate the target of your interest. And we are able to provide you with custom probes, as well as you are welcome to over 26,000 pre-designed probes in our catalog. Because of the sensitivity and specificity of our assay, we can very precisely spatially map targets of interest in heterogeneous tissues. With our specialized assays like microRNAscope and BASESCOPE assays, we can look at targets like non-coding RNAs, point mutations, splice variants, and microRNAs that are very difficult to detect and visualize with other tools. In addition, our assay can be combined with immunohistochemistry and immunofluorescence to visualize RNA and protein on the same section of the tissue. Ultimately, because of the plexing flexibility and the sample type flexibility that we offer, we have been widely applied in different research areas. With that, I would like to welcome any questions that you might have. Please reach out to us with any questions that you may have later. Thank you.
Thank you so much, Anushka. What an excellent presentation that was. While I go ahead and review the questions that we have for you, I will hand it back over to Pupak. Thank you.
Thank you very much. This ends our presentation portion of the event. Before we open up all questions for Dr. Dikshit, I'd like to let you know that we'll have more knowledge resource webinars in the upcoming weeks. Our next topic will be on stem cell applications, being held on September 22nd. So look for our upcoming invitation. Meanwhile, I'd like to encourage you to visit our website at acdbio.com to learn more about our products and services. Be sure to contact our sales and support colleagues if you have any questions or if you'd like to discuss your projects. Thank you. We'll open up with questions.
Great. Thank you very much, Pupak. Okay, we did get quite a few good questions today, Anushka. So the first one came in: is it possible to apply this technique to organoid models?
Thank you, Rhonda. Yeah, that is a great question. I did not show specific examples, but we have been used, our RNAscope has been used to study target expression in organoids and 3D cultures. So yes.
Great, thank you. Secondly, how feasible is combining RNAscope-ish with IHC, immunohistochemistry?
That again is a very great question. I showed a couple of examples wherein researchers have published using RNAscope in combination with IHC or IF, and we have protocols that help you guide how to use these two workflows simultaneously. And we have a document published on our website which shows you a list of over 300 some publications that have used RNAscope in combination with IF and INC, and it can be easily performed.
Great, thank you. Good to know. Another question that came in, will BASESCOPE be available on the automatic platforms soon, or is it available now?
So we have singleplex BASESCOPE available in automated platforms, and based on what tissue type and which assay you're specifically interested in, that can vary, but BASESCOPE is available in automated platform, but singleplex only. If you want to perform duplex, it's manual.
Okay, thank you. So as long as we do BASESCOPE singleplex, that sounds feasible. Great. And obviously, if you have more questions, please do reach out to the support team and they can walk you through those protocols. Another question here, are there any probes designed for the COVID-19 research? Obviously a hot topic right now.
Yes, as from one of the examples that I showed, we have over 25 publications now that have used our RNAscope probes for COVID-19 research. These include probes specifically against the SARS-CoV-2 viral RNA for host cell receptors and proteases, and also for cytokines and chemokines that come up after mounting and after the host mounts an immune response against the infection. So all of these probes have been detailed in the publications. So we have been used extensively for COVID-19 research.
Great, thank you. And for those of you who are interested on more, we have a number of pre-recorded presentations discussing our COVID applications available on under the recorded webinars section of the acdbio.com website. Another question here from the field, can RNAscope differentiate between highly homologous sequences?
Yes, and our BASESCOPE assay is a specialized assay particularly designed for that, wherein because the target sequences that we want to use to distinguish between homologous sequences is a small part of the sequence, we need our specialized assay like the BASESCOPE assay, where we can design probe against that unique sequence that will distinguish between homologous targets.
Great, thank you. And then one last question before we close out today. Currently, what is the limit of multiplexing using the RNAscope technology?
So at the present, we can do up to 12 targets on the same section of the tissue with our HYPLEX assay. And that is available for our fixed and fresh frozen samples.
Great, thank you. Well, this concludes our Q&A session for today, and that concludes our webinar, the Knowledge Resource Series webinar today. So thank you all for joining. Really appreciate the great questions and the close attention to the presentations today. Anushka, that was a fantastic presentation. It was recorded, and so in the next 24 hours, we should have the link available on our website under recorded webinars. And any other information you have, questions you have regarding our technology, please review the website as Pupak mentioned. Also reach out to us at the support team email on the screen, and we look forward to seeing you next month for our next Knowledge Resource Series. Thank you very much, everybody. Goodbye.